Okay, so it's my pleasure to introduce our first speaker, Óscar Castillo, who will be giving a talk on the implications of Hyb-Seq sequencing on the generic delimitation of Helichrysum and related genera.
Hello, everybody. Thank you for the presentation, Karma. I'm going to share the presentation now. Okay. So today I'm gonna talk about Helichrysum related genera. I'm currently doing my PhD on Sonchus, but I started my master thesis on Pseudonaphalium while I was at the Autonomous University of Barcelona, and work with the Hyb-Seq also in the Botanical Garden of Barcelona. So first of all, we have to know who are these, what is the Hyb-Seq, and its name because of the most diverse genera in the clade that are Helichrysum, Anaphalis, and Pseudonaphalium. Helichrysum is the largest genus in the tribe Gnaphalieae, the most morphologically and ecologically diverse, and widely distributed in Africa, the Mediterranean, Macaronesian, Western and Central Asia, and India, reaching its highest diversity in Africa and Madagascar. But we also have Anaphalis, which is fundamentally Asiatic, Achyrocline and Pseudonaphalium that are mostly from America. In general, the difference are in sex ratio number and sex ratio of the florets and number of florets per capitula, but in some taxa there may be some exceptions. Anderberg already pointed out the similarity of Helichrysum to other genera with divided or fenestrated stereom. I will show you what is this. And later phylogenetic analysis showed these genera were embedded in Helichrysum, which is interesting because all these, Anaphalis, Achyrocline, and Pseudonaphalium, are polyploid, and molecular evidence from previous studies showed a putative allopolyploid origin. This is a fenestrated Pseudonaphalium oxyphilum, and it's the thickened basal part of the filary, and we can see the two fenestras, or it's fenestrated or divided. But there are also other small genera that are also in the Hyb-Seq, and since the taxonomic treatment, generic delimitation has been a problem to address, and because of all of this. And we will not go very deep into these other genera, but I wanted to see them a little bit quick. For example, Galeoma, with 2 species, we only sampled Galeomas oculus cati, and then we have Syncephalum, that is interesting because it has the pappus is missing in these plants, or Sthenocline, with 2 species that are quite different morphologically, and even though in the bibliography it's said to have undivided stereom, when we checked for Sthenocline nuloides, it had a fenestrated one, so sometimes we don't have enough knowledge of all these species. And then we have Homeocline, which is a monotypic genus characterized by its pyramidal inflorescence, as you can see in the pouches. So the study of these groups has been limited because of its great diversity and wide distribution. Most of the previous works were based on limited molecular markers and lacked representation of the diversity of the allopolyploid genera. So here we have sampled this genera. DNA extractions were performed and followed by a Hyb-Seq protocol to obtain nuclear and chloroplastic sequences using the COS Compositae kit for target capture. With all of this, we use Hyb-Piper to process the sequences and build phylogenies with concatenation approach with RxML and with Astral for coalescence. And in the case of the chloroplastic, only with RxML. Actually, if you want to take a look into phylogeny and know more about the evolution in the Hyb-Seq, in this paper, you can find nuclear phylogenies built with a similar approach that I'm going to present. And now we will focus on a comparison with the chloroplastic ones that is going to help us to give us another perspective on the evolution of this genera. Here we can see the two trees built with Rexamel and Astral simplified. There are some species that fall out of Helichrysum that fall outside of the Hyb clade, and we are going to focus on the Hyb clade that would be from this on. And for example, for Galeoma, it occupies the same position in the Rexamel and Astral. It's close to South African annual, a clade including South African annual plants. For example, for Umeocline and Syncephalum and Stenocline, the position may change from one to another, but they're always related to the same species in both phylogenies. And in the case of Syncephalum, and Stenocline, the two species of Stenocline are not clustered together, and Syncephalum is close to them. Another Madagascar species of Helichrysum. And then we have the biggest genera in the Hyb clade that are Anaphalis, so Anaphalium and Achyrocline. Anaphalis is close to the Mediterranean Asiatic Macaronesian clade of Helichrysum in both phylogenies. Is there any? I don't know if I'm gonna follow. And then, but it's not monophyletic on the Astral. Also, so Anaphalium and Achyrocline shown to be monophyletic in the concatenated, but with low support. And in the Astral, they are mixed together, they are polyphyletic. And when we see the chloroplastic, we will see deeper on the biggest genera, but for example, Galeoma, it's not anymore in the base of the clade, but it's related to the same species as before. And Syncephalum and Stenocline, instead of go together in the same clades, they are related to, well, Stenocline closer together than before, and in a different clade than Syncephalum. And for Anaphalis, we see that it's split in several clades, the same for Achyrocline in two clades, and for Pseudoanaphalium also. So the story gets complicated when we look at the chloroplastic, as it usually does. So regarding Anaphalis, most of the species are subdioic, but it's not clear for the Indian species or the South American one that we couldn't sample in this case. And it's monophyletic on the concatenated, but not in the Astral, with monogynum and some species of Anaphalis clustering at the base. And when we see in the chloroplastic, we can differentiate three clades, sometimes closer to diploid species of Pseudoanaphalium that are Asiatic, like Pseudoanaphalium chrysocephalum and Pseudoanaphalium hippoleucum. But what is interesting is these clades are, in terms of composition of species, are similar to the ones on the concatenated, well, on the nuclear phylogenies in general. Then we have the Mediterranean Asiatic Macaronesian Helichrysum. This group is interesting because it's the type of the genus, it's Helichrysum orientale, it's in this clade, and it's also of a putative allopolyploid origin, closely related to Anaphalis, as we've seen. And what is more interesting is the fact that Helichrysum monogynum, which is an endemic of the Canary Islands, Lanzarote, it's not clustered to the other Macaronesian species, but it's in the base of the clade of Anaphalis and Mediterranean Asiatic Macaronesian Helichrysum. And in the chloroplastic, it goes with Eastern African species that they are not anything related. This with the fact that it's an hexaploid suggests that it has a more complex evolutionary history than the other species on this clade. We also have here Achyrocline. Here we can see two species characterized by its narrow full-fledged capitula. Helichrysum stimperi actually was transferred to Helichrysum, but we will see it's closely close in the phylogeny. And also I will treat Gnaphalieae together because it's considered a synonym to Achyrocline. In the nuclear phylogenies, since Gnaphalieae is always embedded in the same clade, we can differentiate two clades of Achyrocline, but they don't have the same composition in the concatenated and astral, even though sometimes the super values are not very good. But they are close. What is important is they are close to Helichrysum stimperi and Helichrysum sordidum. Instead, when we look at the chloroplastic, the two clades are separated and not close to stimperi or sordidum, Helichrysum sordidum. And the last but not the least, Gnaphalieae. Its genus include few diploid species, but most of them are polyploid and grow in America. In the XML, we can see two big clades with most of the polyploid North American species and most of the polyploid South American species. But we can also identify two clades with diploid species from different regions in the world, and also Helichrysum Nicolás from Cabo Verde, which has a florent sex ratio that is of a Gnaphalieae. So it makes sense. And here we can see that the Achyrocline would be close to Gnaphalieae, making it not polyphyletic. And when we look at the chloroplastic, we can see again the two big clades of polyploid species, this time with an diploid African species that is Gnaphalieae undulatum at the base, and Gnaphalieae viscosum, the only diploid species from America, close with the North American species. This, together with the other Gnaphalieae that are diploid in the chloroplastic, that are close to the Asiatic Anaphales, gives the idea that here there's some kind of geographic pattern that we are seeing. And the clade of Luteal album, including other diploid species. So now, what options do we have? What can we do? When we look at the bibliography, the taxonomic proposals are often based in three pillars: monophily, completeness, diagnosability, and nomenclatural stability. That is, that the genera should have diagnosable characters, species should be easy to assign to a genus, which has to be monophyletic while avoiding too much nomenclatural changes. So one possibility could be to split Helichrysum in small genera. That way we could recognize allopolyploid groups, but this would involve a huge number of name changes. We would need to find clear diagnostic characters for each new genus, which is difficult because some groups are highly variable in their morphology, while others look very similar to each other. In addition, placing unsampled or poorly known species into this genera would be not easy, and it could even lead to the creation of more genera in the future. Overall, instead of improving nomenclatural stability, this approach would probably make the classification even less stable. Other option, if we want to avoid a lot of new combinations while preserving the identity of lineages, would be something similar to what we have now. So a paraphyletic Helichrysum. This is difficult to defend, but if we want the nomenclature to reflect evolution, we have to consider that allopolyploid genera have more in common than with other Helichrysum. And not having a proposal to the African lineages of Helichrysum does not mean that the polyploid lineages do not have a particular identity. Also, maybe we should question monophyly as a criteria. At this point, allopolyploid groups will always have a fake position in a phylogeny. We cannot represent reticulate evolution in a dichotomous tree, and I think we should focus on its origin, evolutionary history, and isolation from other lineages. Evolution often implies paraphyletic phases, and we just have a picture of it. Anyway, if we wanted to recognize allopolyploid groups and avoid many nomenclatural changes, we should accommodate the Mediterranean Asiatic macaronistic species, which are polyploid, in a new genera, and change the type of Helichrysum to an African species. That is difficult to define, and given the morphological exceptions in allopolyploid lineages, the complicated evolutionary history, and the presence of diploid species in Pseudonaphalium, it doesn't seem a really good option. And the third option, lumping everything into Helichrysum. It would imply a lot of nomenclatural changes, that's true, the loss of traditional accepted genera, and make Helichrysum a super diverse genus present in other continents like America, where it wasn't. And it's against the trend into other Gnaphalieae that tends to make smaller genera. But it follows similar approaches to other big genera like Salvia or Carex, for example. The assignment of the taxa would be easy, everything would be Helichrysum, and fenestrated Asterium would be the evolutionary shared feature, even though sometimes can be lost secondarily. So comparing these possibilities, and after all these considerations, nowadays we are working on the combination of this taxa in the hub clade into Helichrysum. So thank you for your attention. Thank you for all the members of the group that made this possible, and I hope you like it. I don't know if I can wait. All this. Sorry, I'm a little bit.
Thanks for that great talk, Óscar.
Uh-huh.
We have time for a short question, maybe. Anyone would like to join? Feel free to unmute and ask a question. Yeah, go ahead, Nicolás.
Hi, yeah, thanks. Thank you, Óscar, for presenting very, very complex results so clearly. Yeah, it's indeed a difficult question. I know it's something that you've wrestled with a lot. It's not easy to choose a path when there are problems with every choice. So the one thing I was just wondering about is, given that there are so many polyploids, do you think that there would be more insights if one somehow could take that polyploidy into account in the data analysis? These are not methods that I've done myself, but I believe there are methods for bioinformatic methods for Hyb-Seq data with triploids, for example. And I mean, I don't know, I don't think you'd necessarily be able to bring out the different subgenomes, but I just wonder if it wouldn't help something like that, or the other possibility would be some kind of phylogenomic network approach if they're known allopolyploids.
Yes, for sure, networks would be very useful to all these allopolyploid problems. I think now the problem is that hub clade is so big that to make a phylogenetic network, I don't know if it's feasible. Maybe with some subsets of the sampling, including all the lineages, to try to elucidate this. And this, I mean, there's a lot of work with the origin of all these allopolyploids. I read a paper on allopolyploids in Stachys, Aplostachys and genera that are endemic to Hawaii. It was published in Nature, and they used they have a reference genome, and they could do some more sophisticated analysis, and with these hemigenomes being represented twice in the phylogeny, so you could match one hemigenome to one lineage and one to the other, the parentals, which is what is bringing confusion on these trees. And this is something that would be really interesting and will help with all of this. But I think the problem on genetic delimitation would still be there to some point, because even we can know about the origins of Pseudonaphalium, and Anaphalis maybe has like multiple origins, but maybe with the same parental lineages, we will still have the problem that they are difficult to fit in a dichotomous tree, and they have great morphologic variation, because not all Anaphalis is subdioic, or not all Zonophalliums are polyploid, so they must be need to study Zonophallium and Achyrocline anaphylis in separate analysis, and even the Mediterranean, obviously, clade, because it's very complex. And in this paper on Stachys, I remember that they did all of these new methods, and they quite arrived to the same conclusion that using a chloroplastic and a nuclear phylogeny compared. So, because sometimes it's difficult to know if there's been one event of hybridization or several events from close lineages. But I would like to try to do something like that in the future, for sure.
Mm-hmm.
I don't know if this...
No, but you make some very good points. Yeah. Thanks.
Okay, we should move on to our next speaker. I'm happy to introduce Dr. Juan Manuel Gorospe, who will be giving a talk on the use of integrative taxonomy to assess the diversity of Oritrophium in the tropical high Andes.
Hello, can you hear me?
Yes.
Yes. Okay, thank you, Karma. I will share my presentation with you. Can you see it?
Yes, you can.
Okay, perfect. So I will talk today about a study focusing on Oritrophium sensu stricto, which was part of my PhD thesis and also a student project that I had back then. So the genus Oritrophium belongs to the Asteraceae subfamily, and then it has a rosulate habit with escapose and monocephalous inflorescence, in which these florets are functionally male. It has around 23 described species, but it has also a complex morphology. At the beginning was assigned to Aster, then was part of sections of Helichrysum or Selmisia, and then in 1961 it was finally assigned as a different genus by Quatre Casas. Here you can see some of the representatives of the tribe, which grow at high elevations, usually in wet and swampy habitats. And these are photos from the Paramos around above 2,500 meters or 3,000 meters in Ecuador and Peru. The typical habitat would be more these ones on the right side, growing close to the water, but then in these ones on the left, you can also see some patches of more swampy habitat or moist habitat, which you can also find some of these species. It grows all along the Andes from Bolivia, which you can see here on the map, up to Venezuela. And there's also one interesting disjunction with two representatives in the alpine zacatonales in Mexico. It would be up here. But most of the diversity is found from the north of Peru to Venezuela, and it grows in the páramos, which would be more or less the black habitat or areas highlighted in the map. And this isolation distribution makes it very interesting to study evolution. Here I wanted to highlight some of the main hypotheses of the origin of the alpine flora in the Andes in general. So first, the origin is usually referred or explained by the major Andean uplift from 10 to 5 million years ago. And a bit later, the emergence, the páramo emerged, and then this availability of new habitats allowed for colonization from taxa from the mountain forests, and also from high elevation habitats in temperate regions. The main hypothesis for the diversification of this flora is linked to the Pleistocene climatic changes, which influence the flora during warmer conditions. The populations were confined to the top of the mountains, being isolated there, and during cooler conditions, the glaciers grew, and these ranges are hypothesized that migrated downwards, forming corridors from one peak to the other. And then this, if you imagine a constant cycle of isolation and connection, favored scenarios of allopatric speciation, secondary contact, hybridization, and also migration between mountain peaks, triggering the diversification of many Andean genera. Going back to Oritrophium, one study in 2022 using Sanger markers, some plastid and the ITS, showed that Oritrophium, as currently circumscribed, is polyphyletic, with 5 species that I highlighted here in red, being that must be assigned to other genera, some to Helichrysum, some maybe to Diplostephium or Hintubera, and but this is work that needs to be done, and this leaves Oritrophium sensu stricto with 18 species that originated in the early Pliocene, so linked to this origin of the páramo. They obtained a good resolution for most of the species included, which was almost all except a few very narrow endemics from Venezuela and one of the Mexican species. But they didn't obtain a good resolution with enough resolution for the Oritrophium peruvianum group, which would be here. And here I want to show a bit the distribution of the Oritrophium sensu stricto, and as you can see, we have 2 widespread species Oritrophium peruvianum in green, here on this map, and Oritrophium lignophyllum in yellow. And then we have Hyb-Seq, which also a distribution from Peru to Bolivia. And then we have species with narrower distribution and a lot of endemics. After that, we need a Hyb-Seq phylogeny using the Compositae probe set, and using almost 950 loci, we obtain again a good resolution of Oritrophium, but still we didn't find a resolution for the Oritrophium peruvianum group, shown here in green. And interestingly, we included one sample of Novenia, and the taxonomic would need to be revised, hopefully including the other Mexican species to really see if this relationship is correct, because Novenia has a lot of it doesn't resemble so much Oritrophium as it does Oritrophium orizabense, but here we see it within Oritrophium, and we include it because there was some another study based on the tribe that already highlighted this relationship. Anyway, we would focus on the Oritrophium peruvianum group today, which I show some representatives here in the pictures, and the taxonomic differentiation of each species is based mainly on vegetative characters, mainly the leaf shape, the leaf margin, and the type of indument. And the boundaries between each of these species, which maybe you can already see or get a hint from the photos, overlap to some extent, making it sometimes difficult to assign a particular individual to one or another species. So based on this, and on the low resolution of the Hyb-Seq clade, we asked if we could find support for each of these species based on different species concepts. We used three. The genetic concept would identify distinct genotypic clusters. The phenetic concept being able to also find clusters based on different morphological characters, and an ecological concept which will imply that the species occupy distinct adaptive zones. For the genetics, we sampled 235 individuals from 38 populations. We included all described species from the group, and we covered all of the distribution range, focusing on the central and southern part of Ecuador, which holds most of the diversity. We applied Hyb-Seq on these samples, which allows you to get a good representation of the genome, and is useful for taxa that don't have a reference genome. And this allows us to get polymorphisms. And after processing the data, we got around 2,500, a bit more, to use for analysis. The phenotypic criteria involved recording characters of 54 herbarium specimens with more than 300 individuals. Again, we measured all described species and cover the distribution range of the group. And we measured 14 vegetative characters and 41 reproductive ones. After processing the data and taking out some characters that were too descriptive and couldn't be analyzed, we were left with 362 individuals and 50 characters. Lastly, for the ecological criteria, we went to our records. Also, we took those from GBIF that had coordinates within the Páramo and were identified by people known to be specialists in Asteraceae or Páramo flora, and we combined them with worldwide environmental variables from climate and soil. Looking at what we got for the genetic criteria, I show you here a distance-based network in which you can see that the species that are represented by the colors are not supported, and rather we have more of a geographic pattern. We did also some admixture analysis and we got similar results, but I usually show this one because it's much more easier to see. For the phenotypic criteria, we also were not able to easily distinguish the species clusters. It's true that here Oritrophium tergoarbum in purple could be a bit different, but then this axis only explains less than 10% of the variability. For the ecological criteria, we analyze the data using hypervolumes, which builds the needs in 3 dimensions, and calculating the overlap, we saw quite a lot of overlap. Also, in some cases, it was difficult to estimate the needs because some species are very narrow endemics, and also using worldwide climatic data maybe is not the optimal resolution, but it was the only data available for us. So based on this lack of support, we decided to change the perspective and focus on the speciation process and on the continuity of the process of speciation, which I try to show here. On the left, we will have one end of the speciation continuum in which we have low reproductive isolation, and the diagram up here is trying to represent a divergent process. So we would at this end have something like 2 populations that can still easily reproduce, but maybe are showing some differences. But we would consider them one same population or one same species, depending on the level in which we are working. In the middle, we would have more differentiation, but perhaps in some criteria we would see a more clear differentiation, and in another we would not. So maybe we would see some morphological differences, but maybe the ecology of the species is the same. And at the other end, we would have high reproductive isolation, and we would consider the 2 populations, in this case, different species. So based on this speciation continuum concept, there was proposed the speciation curve, and later the speciation Hyb-Seq, in which you try to combine different criteria to reassess if each population pair, and it's important that here we are comparing different populations. Each population pair is at one or another placement in the speciation continuum. So here we could integrate together our genetic, phenotypic, and ecological criteria, and here it would be where the three examples that I showed before would fall into this cube. So the populations that were at the lower end of the continuum would be placed here, so they would show likely low differentiation based on each of the three criteria. The intermediate case could fall around here, and then the two distinct species would show differentiation across all criteria, and could lay somewhere around this area. To explain a bit more about this, I bring you some examples from the paper in which they describe this speciation Hyb-Seq, and these would be just theoretical examples. Then you work with the data, usually you get a different pattern, but this pattern would resemble divergence in allopatry. So you would start to get genetic divergence together with phenotypic divergence, and slowly reproductive isolation would start to increase. Divergence in parapatry, for example, would have a high differentiation in phenotypic traits as well as in reproductive isolation, and at some point species would differentiate genetically. And here at the bottom there is examples of reinforcement after secondary contact, and when the secondary contact reverses the speciation process. So to apply this, we had to combine all of the different criteria we had, and we decided to include some more. But this meant that we needed to reduce the number of populations to just 13. But still, we were able to include most of the species within the group. And reproductive isolation, we had to estimate it as a proxy because we couldn't perform crosses, and we estimated current inflow based on our SNPs. Genetic differentiation was based on FST. And then for the phenotypic differentiation, we, as we had different individuals from each of the populations, we calculated the centroid in the PCA, and we estimated the distance to reflect this differentiation. For the ecological criteria, we split the climatic and edaphic differentiation, also estimating the distance between the points in the PCA. And lastly, we included the distance between geographic localities. To compare all of these criteria, we normalize the data from 0 showing a low differentiation or identical population to 1 showing distinct populations. This would be our speciation Hyb-Seq, in which we see that population pairs, represented here by gray dots, are all across the plot. So this indicates that they are following different trajectories across the speciation continuum. Some populations are differentiating more based on the geography. Some are explained better by the phenotypic differentiation, and so on. And then if we select 3 criteria to build a cube, we see that roughly at one end of the speciation continuum, we have some populations and some others fall at the other end. And this pattern, while being quite different, it resembles a bit the allopatric scenario with more complexity, of course. And to clarify a bit, if we could find support for species, I highlighted some population pairs, some examples here. So, for example, the red dots would be comparisons between populations from the same species. The yellow would be a similar case, and the gray would be comparing different species, but which are usually very difficult to segregate sometimes based on morphology. And you can see that these tend to fall at the lower end of the, or more at the left and bottom, with some outliers, of course, highlighting also already the complexity of this analyzing the process of speciation. But then, for example, the cases in orange, which is Oritrophium younganatense and all of the other population, rather fall at the other end of the speciation continuum. So if we try to place the different population pairs and look at which is the assignment of each of these populations, we can more or less construct a continuum in which in one end we would have species that are assigned to the same, individuals or populations that are assigned to the same species, and at the other end, populations that are narrow endemics. And I want to look at the connectivity in the Andes, and I saw that for the case of Ecuador, which is the center of diversity for the group and which has most of the narrow endemics in the Oritrophium peruvianum group, the south, which is the center of diversity, has less connectivity in the last 1 million years compared to, for example, regions in the north, which maybe we have one or two more widespread species. And also the plot here is showing how the connectivity changes through time from past to present. And we see that it had a lot of fluctuations, which would imply that there were all of this process of isolation, then secondary contact, migration, etc. And this could explain why we see some populations being different, but not in all of the criteria, and why some others, for example, maybe from more northern localities, are more clear that belong to the same species. So with this, to sum up, we saw that the spread species within the Oritrophium peruvianum group were not fully supported based on species concepts, and likely they represent heterogeneous speciation stages and trajectories across a speciation continuum that could be associated with the geographic isolation and secondary contact processes resulting from the Pleistocene climatic changes. Well, if you are interested in this study, it was published last year, in molecular ecology. So I hope if you go to it, you enjoy reading it. And just thank all the people that were involved in the study. Also thank Carmen and TICA for the opportunity to present this work to you. Thank you very much.
Thank you, Juanma, for this very interesting talk. Since we are a bit short on time, I'm afraid we need to move on to our next talk, but I encourage you to continue the conversation in the chat or by email. And now we come to the final talk of the session. Please welcome Nicolás Ramírez with a talk on the taxonomic revision of Achyrocline in Colombia.
Hi, can you hear me? Okay,
Yes.
I'm going to share the presentation. Let me... can you see it now?
Yeah.
Okay, so if you heard the previous presentation, Óscar's presentation, the genus Achyrocline now is going to be Helichrysum. So today I'm going to talk about Achyrocline or also the few-flowered Helichrysum of Colombia. So Achyrocline come from the Greek words achiron, meaning chaff, and kleine, meaning bed. This is because when fruiting, those species practically become a mess of filaries and flowers that look pretty much like chaff, as you can see on the right. The genus was described by Lessing in 1832, but as a subgenus of Gnaphalieae. But in 1838, by Decandol, it was elevated to genus level. This genus is widely known as Viravirus all across South America. Well, as previous presentation mentioned, this genus belongs to the Hyb-Clade within the subtribe Gnaphalieae and is included within Helichrysum. But this genus is mainly an American genus growing from Mexico to Argentina and Chile, and it has like around 44 species in total. But in Colombia, only 9 species were reported in 2016, and the highest species richness belongs to Brazil with 22. So, how to recognize this genus? Although many of these plants may look like herbs, they are actually subshrubs, perennial subshrubs that have a lignified base in their stems, and they are highly aromatic plants. They smell like turpentine. But the more important characters to recognize this genus are in the flowers and in the reproductive parts of the plant. The most important one is they have few flowered capitula, less than 10 flowers, and in Colombian taxa, mostly 5 flowers. The pistillate flowers outnumber the bisexual florets, and the stylar arms are truncated at the apex, as you can see on the left. Also, the capitula are deciform, so they have pistillate florets and bisexual florets. As you can see on the right, they have a divided asterium, that is this structure here on the filaries, and they have glabrous and smooth cypsellae, as you can see on the left. It's sometimes easy to confuse this genus with Pseudognaphalieae. quite easily. But although there are so many examples or characters that you can use in the field to differentiate those two genera, the more important one and the only one that can differentiate these two genera is the number of florets. Pseudonaphalium has more than 20 florets. That's the basic one, the most important one. And in the field, some people might confuse this genera with other genera within the tribe, but the reproductive characters mentioned before are the most important to differentiate Achyrocline from these ones, especially the Indumentum montecipsela and also the Asterium montefiloris. And something really important about this genus and why most literature focus on is the medicinal properties. They are used all across South America by communities to treat gastrointestinal and urinary illnesses. That's why it's highly studied in the pharmacology due to its active compounds. But although it's really uncommon, it has a decorative use. You see, some people use this in dry floral arrangements and crafts. And in Colombia, it grows especially in Páramo and some subxerophytic Andean environments, like you see on the left, a Páramo in Colombia. Previous work about this genus is just mainly local floras all across the continent, starting from Argentina to Mesoamerica. So in Colombia, we lack a full taxonomy revision and of this genus, and we are highly in need of one because there is a lot of confusion of the identity of the species in Colombian herbaria and also in literature. Some literature, especially in pharmacology, tend to confuse the species they are working on. So this is why I'm working on the revision of this group. I check around like more than 1,000 specimens all across 18 Colombian herbaria. They did several field trips to Boyacá and Cundinamarca, mainly Western Cordillera of Colombia, and also check the protologs in JSTOR and BHL. This is all the herbaria I checked, mainly in COL, the National Herbarium of Colombia. This was the most important one and where I worked. So in Colombia, this genus is distributed all across the Andean region, all across the three cordilleras, the Eastern Cordillera, the Central Cordillera, and the Western Cordillera, and also the Sierra Nevada Santa Marta, and it grows in a range between 1,000 and 4,000 meters above sea level. But most species grows, especially above 2,000 and below 3,000. So now we are going to see the diagnostic characters. What characters are useful to differentiate the species in Colombia? All of those are vegetative characters because reproductive characters are really not useful to differentiate species. But all species across this genus has reproductive characters pretty similar and highly variable, and they do not follow some patterns, so they are not really useful. So when you see the vegetative characters, you can see differences, consistent difference. Some species, well, in Colombia just one, which is Achyrocline alata, have the leaves are decurrent on the stem, and that's why you can see winged stems compared to some species like this one that the stems are wingless, right, practically. Another, I will consider this like one of the most important leaf characters to address this genus, is the leaf shape. Some species have a pseudopetiole, a highly conspicuous pseudopetiole, like you can see here on the left. But some of them don't have this. So this characteristic, the presence or the absence of a pseudopetiole, is really useful to differentiate species in this genus. Also, the basic shape of the lamina, like some species has linear linear leaves, and some others have more like a lanceolated one, or sometimes like ovate, as you can see on these ones. Another character, and I will say it's not easy to see this character, is the presence of a mucro. So in the apex of the leaves. So if you can see, like in the right one, you can see a tiny bit of leaf, like it comes out from the indumentum, and this is the mucro. But this is absent in some species, like the one on the left. But this is really difficult to see because you have to peel off the indumentum just to see this character. In the same, also in the same sense, there is a difference in the margin of the leaves. Some species have a flat margin, and other ones, like the one on the left, have a highly revolute margin. But again, this is only visible when you peel off the indumentum, so it's not really easy to see. You have to really dissect one leaf to see this character, but it's highly consistent. And another character, the indumentum, but not like the type of indumentum, because many of the species in this genus have like a woolly indumentum that can be lax, like maybe arachnoid, or really dense. But the important difference is if the leaves are discolorous, like here in these ones, or they are concolorous, like the one on the right. Also, some species tend to have like a lot of glandular trichomes on the underside of the leaves, but the presence or the absence is not really a taxonomic character. It can vary from one specimen to another, and this is why they smell a lot, even when they are dry. Another character, and maybe the only reproductive character that may be useful to differentiate the species, is the shape of the inflorescences, the synflorescences. It is because, well, the basic inflorescence in this genus is like a glomerule, like a cluster of capitulum. But some species tend to have like solitary glomerules or few glomerules, like in the left one, and other species tend to have like very big panicles of a lot of glomerules organized very loosely. So you can see that on the right. It's sometimes it's not that easy to establish which one it is, but generally is kind of useful. So with these characters, we were able to differentiate two different groups within Achyrocline in Colombia. One that has pseudopetioles, flat margins, mucro, and big inflorescences. And one that has like that don't have a mucro, the margins are revolute, and the inflorescences just has like solitary glomerules or clusters. And the leaf bases are semi-epiloxical. They don't have a pseudopetiole. Those two groups are the basic morphological groups present in the country. And these are some of the species present on both of those groups. One of the groups here, I call it Group A, have 7 species, and the second one, Group B, has 4 species. Well, as a result of this taxonomy revision, we were able to find 2 different species. Both from this species group B that have revolute margins, and both endemic to Colombia. One that we could find like fresh material, like in field trips, and the other one only known from a herbarium specimen for the 1940s that doesn't have flowers, mature flowers. So those species one of those grows in the Western Cordillera. For those who do not know Colombian geography, this is the Western Cordillera, and is the red dot high in the department of Santander. And the other species grows isolated in the Sierra Nevada de Santa Marta, and you can see that in the green dot. Also, there are some novelties, and this species, the species on the left, Achyrocline celosioides, is a new record for the country. It was already reported from Venezuela, from Ecuador, from Peru, and Bolivia, but Colombia was missing, and we could find just a single specimen of this species on the left, because it was actually mixed with another species of Pseudognaphalieae, so the one on the left. And also, although it is not exactly a new record, Achyrocline vargasiana, it was before considered a synonym of Achyrocline satureioides, but now here is considered like a different species, Achyrocline vargasiana, present also in Colombia. Among some other novelties, the one on the left, Achyrocline remotifolia, is also a new record for the country, and it's because this species was described as a subspecies of Gnaphalium satureioides and considered a synonym. So this species is now elevated to species level. So now here is considered a different species apart from Achyrocline satureioides. And on the right one, also, we have a new synonym. This species described by Clad is now considered a synonym of Achyrocline bovotensis. Also, we have some excluded species. These species were described from Colombia. There are only known from the type specimens, and they do not belong to Achyrocline in the strict sense. Maybe they are actually Pseudognaphalieum, but no species in Pseudognaphalieum. They are actually synonyms of some species already existing in Pseudognaphalieum. So these species were excluded from this work. Also, there are a lot of lectotypifications that they need to be done, because, for example, some of the type material were destroyed during World War II in Germany, in the Berlin Herbarium. And in other cases, some of the original descriptions cite two different specimens as the type. So there are syntypes that need to be lectotypified. There are actually 5 lectotypifications. So in summary, there are actually 11 species present in the country. 2 of them are new. One of them is a restatement. Actually, it used to be a synonym. There's a new combination. There's a new synonym. 2 species were excluded, and there are 5 lectotypifications. So, thank you. That's all. I have to acknowledge, especially the institutions that helped me with this work. That was my university, Universidad Nacional de Colombia, and the Instituto de Ciencias Naturales, where I work this research.
Thank you very much, Nicolás. This was a great talk. I think we'll have to wrap up here today due to the time. So this brings us to the end of today's session.